Hello everyone, I am Ishak Mondol. Today I will explaining a bioinformatics topic. The topic is pairwise sequence alignment algorithm, and we understand in this video the algorithm of local and global alignment. So let's start. At first, what is sequence alignment? Sequence alignment arranges two or more sequences to identify a region of similarity that may indicate functional, structural, and evolutionary relationship in the sequence. Here is an example of two sequences. Then, what types of pairwise alignments available? In this pairwise alignment, mainly two types: global alignment and local alignment. Global alignment is aligned to entire length of two sequence from start to end, and local alignments to identify the most of similar region between sequences. An application of global alignment is full sequence comparison, sequence of similar length and high similarity. In local elements, finding the conserved domain and motifs, detecting the functional regions to distant related sequence. What is the key characteristics in global alignments? It's optimized alignment over the entire sequences. And local alignment is the find the highest scoring local segment. In global alignment, we used algorithm is Needleman Wunsch. In local alignment is used Smith-Waterman. Global alignments. The global alignments it aims to align entire sequence from start to end, making it ideal for sequence of similar length and high similar. And it is used to Needleman-Wunsch algorithm, the classic dynamic programming to algorithm global alignment. And in global alignment, to use the edit distance to measurement the minimum numbers needed to transform one sequence into another. Here the example of global alignment, it is ideal for the similar length of sequences. The global alignment algorithm is based on Needleman-Wunsch algorithm, global alignment. So in Needleman-Wunsch algorithms is the classical dynamic programming method. To Needleman-Wunsch algorithm construct a scoring matrix where each of cell represent the optimal alignment score substituting the position. So it is a recurrence relation of this each cell where each score put, and it traceback begin in FN positions to follow the path of minimized score. How to implementation of the Needleman-Wunsch algorithm. First, matrix initialization for sequence A and sequence B. Then recurrence relation, how to score predict of each cell in up, horizontal and diagonal. It is the three recurrence formula to how to score put in each cell, and traceback procedure in Needleman-Wunsch algorithms to construct alignment start from FNM, move the cell to contribute it to score, repeat until to F00, align if the diagonal gap is above the left, and gap opening and gap extension panel are separated. And in case of local alignment identify the most of similar region between two sequences, and it is a key difference of the global alignment to find the highest scoring local segments, resets to negative score to zero. Here, in case of global alignment, it is not zero, it is the negative score. In the start test, the highest scoring cell of matrix. And local alignment is based on Smith-Waterman algorithm. And the Smith-Waterman algorithm identifies the most of similar region between sequences regardless of the list of sequence. What is the key features? Uses zero reset strategy during matrix filling. Traceback start from highest scoring cell, ends when zero score reached, and it is a recurrence relation from how to score predict of each cell in when we predict our local sequence. And Smith-Waterman implication, how to fast over matrix construction. Initialization first row is zero, and field using recurrence relation. Here's the recurrence relation. It for its diagonal, for it is vertical, and it is the horizontal. How to trace by procedure? it is difference in difference case of local element. To traceback procedure, the start the high scoring cell move to adjacency to high score. It is the high score in the cell matrix, and it is the traceback procedure how to score it defined to local elements. And the Smith-Waterman algorithm can extend to affine group score to better model the biological reality. It is different gap opening instantly and more realistic for biological sequences. And algorithm selection, why in case we see that algorithm and how to predict to it is global or global or local to function. This is the used for local for discovering the conservative region when we, and used for global how when we comparison to similarity and high to sequences. The application of the pairwise sequence elements algorithm in genomics, in proteomics, and database searches, and whole in genomics, whole genomes alignment for variant discovery, assembly, and comparative genomics. In proteomics, protein structure prediction, functional annotation analysis. In database, to large scale sequence for functional annotation discovery. What is the future direction? Algorithm selection, future optimized, reducing the optimal complexity of the real times in machine learning and in cloud computing. And thank you for watching video.